Let's pass the mic around. Do you guys have a mic over there? Yeah. Okay. Let's just do an introduction for those that might not know you guys, and kind of just say a little bit about your background as well.
Mm. Hey everybody, my name is Jordan Jacobs. I'm coming here today from Portland, Oregon. I am a amateur mycologist and chemist. I study mostly psychedelic mushrooms. I also do a lot of foraging of um, medicinal and edible mushrooms and just cool mushrooms. I take pictures of mushrooms. I spend a lot of time looking at mushrooms. I also lived in Northern California for 7 years, so I spent a lot of time in the cannabis space, making extracts and concentrates and growing and trimming and yeah, just generally interested in mycology and chemistry. I currently run a mushroom analytical lab um, where I test mushrooms that me and my friends grow for our own curiosities, and then I also test mushrooms for other people. So, yeah, a little bit about me.
What's up? My name's Steeg. I just like mushrooms. I like growing mushrooms. I think I take a more like philosophy type approach with mushrooms versus like science based. I definitely like the science, but I don't always understand the science. So I'm actually hoping to learn a lot while I'm sitting up here. Because I think I talk about things in a little bit of an esoteric way, but that's just how I know things. Um, but I think my main passion is just everything non-cubensis and trying to grow as many psychoactive mushrooms as there are in the world and keeping up with the new discoveries and trying to get my hands on whatever, like a Pokemon trainer, collect them all. And also trying to learn how to do mushroom breeding currently, which is an exciting endeavor. So, yeah, that's a little about me.
Hello, everyone. My name is Emily. I'm coming from Brooklyn, New York, and I am an environmental scientist. I study soil and I work in soil remediation. I also am a founder and educator with the Center for Mycological Analytics, which is a project that I've been working on with Ian Bollinger, if any of you guys have heard that name before. And we have kind of been going around and creating pop-up labs to teach people how to do mushroom testing in a very like intensive manner. So it's almost like a 3-day college crash course, honestly. Um, so yeah, that's a little bit of what I do.
Hey, what's up, y'all? My name is Tony Sinius. I'm a mushroom enthusiast, cultivator, forager. I just love mushrooms. I love growing them. I love eating them, and I love how they just bring people together. Um, I my project is the mushroom block. Um, we teach people how to cultivate mushrooms and forage them, and just connecting people with nature of that sort. And I think that's just about it. I just love mushrooms. I just think they're tight.
Awesome. Okay. So, well, I'll tell you a little bit about what we're going to be talking about today. So it's going to be kind of a little bit of a whole glimpse of like what you can do with fungi, some things that might inspire you to get involved in different avenues from, what we're going to start with. What is a mushroom? What is like breeding? What is, some of the supplementation aspects of it? What should you look out for? That sort of thing. Um, some of the DNA barcoding, sequencing. What is that exactly? Um, we'll also talk a bit about HPLC testing and a little bit of like what they're testing for, that sort of thing. So yeah, I guess one of the first questions I have here is like, can one of you guys just answer like how, what is a mushroom? How does it reproduce? And like, how does it go through its life cycle of working in the environment? And maybe you can explain a little bit, maybe like what's mycorrhizal mushroom, a saprotrophic mushroom, and parasitic. Like what would be those differences?
Does anybody know what a mushroom is? Mm, Um, Well, a mushroom. Interestingly, I knew a mushroom once. Um, the mushroom is. I was just saying to some other folks, to a tree, it's sort of like the apple, right? The mushroom is the fruit um, of the fungus. The fungus being the overall organism, which also has a whole other part of its life lived as mycelium, an underground network of cells that are, depending on which type of fungus, either consuming or breaking down things for nutrients. Um, and just like when you pick an apple from a tree and it has the seeds, the mushroom carries the spores for the next generation. Um, is anyone else? What would you add to that? What else do you think of the mushroom philosophically?
Shit. Man, you got me on the spot. I mean, I think of mushrooms as like little people. I think of them as kids. I think of them as my mom, my grandma. I mean, sometimes I'll grow a mushroom and be like, oh shit, I see granddad in there. You know, it's a little bit of everything. I like to like really personify nature and personify um, mushrooms in general because I mean they had such boisterous personalities and um, I guess I like to look at it from that lens because it's harder for me to like be extractive when I'm treating it as like a friend or a family member type of thing. You know, like I'm not gonna fuck over grandma type of situation. um, so I think in that sense, mushrooms are like harbingers of life. They're kind of an expression of repopulation, an expression of gratitude, an expression of the, you know, like kind of think of like how birds do mating dances and shit. Like the way that a mushroom expresses itself is like kind of like a mating dance in a sense, and just like this beautiful expression to the world. Like, hey, look at me, I exist. Like, think about what I do, and like this is how I'm gonna express that to the world. So I look at it in that lens as a mushroom to me.
Yeah, just going off of that. So, mushrooms, they sporulate. And so, when you look at a mushroom, there's like so many different types and kinds and expressions. And they all kind of exist with, like, they all have their own superpower, and that's kind of another conversation. Um, but B, based on their morphology, they have a different way that they can spread their spores out into nature. So like who forages in here? Everybody likes to get outside and look at them. So you guys might know about the puffball mushroom and specifically the one that has that is like a little ball and it has a hole in it and then you squeeze it and then you watch all the spores come out. That's one way that the mushroom can, you know, spread its spores and go into other places. And then we have people in here that are probably familiar with cordyceps mushrooms. Anybody familiar with cordyceps medicine? Nice. So the cordyceps, that's like what the show The Last of Us was based off of. So that's what he had mentioned before as a parasitic mushroom, which it will take over a host, which can be a pupa or, another organism, and it will actually start to like control the host a little bit. And for example, it might make the host crawl up a tree, and then when it gets to the top of a tree, the mushroom will sprout out of its head and then start dropping spores from that like higher place in the forest to the other, locations. So when we look at the mushrooms, like the gills or whatever other characteristic it has, it's looking for a way to propagate itself, and they do it by such like beautiful and unique design. So yeah, every mushroom is just a little like mystery box waiting to be unpacked, I think.
Yeah, I guess I mean, can you repeat the question, please? Please.
How about you? Can you explain what a decomposer is?
Yeah. Oh, that's actually okay. Yeah. So to me, or a decomposer, I mean, like, so these mushrooms straight up are decomposers, and what that means is they break down shit. So like without mushrooms, um, so a lot of these mushrooms actually break down like wood and shit. Like, say again? Of mushrooms? I mean, what's a good one? Yeah, like turkey tail is a good one. Like a lot of these like wood lover mushrooms. I'm just like a little baked right now. Um, Chicken of the woods. You know
Almost
that?
like anything you've eaten before from the grocery
Yeah,
store is growing on wood.
yeah, but I feel like there are some that don't grow on wood type shit.
Yeah. Oh,
Yeah, so
definitely.
that's kind of what I was
Oh, they're
going for.
decomposers as
Yeah,
well. Well,
yeah,
so
yeah,
there's multiple decomposers, right?
yeah. But pretty much, so these mushrooms, these mycelium masses actually break down like shit like wood and other materials. Pretty much. That's, that's why. Oh no, that's a little something different. Just using mycelium as to mold skateboards, but that's a whole nother topic too. Yeah, we're able to utilize mycelium for art, and I mean, people are using it for roads and homes and other building materials and stuff like that. It's really, really interesting. I don't know if that answers your question.
It does. It does. Yeah.
Sweet.
So that's a little bit about. Yeah.
I always think about how like the world as we know it wouldn't exist without fungus because there's so much organic matter that needs to be broken down in order to like propagate these death cycles. And so I really see mycelium and fungus as like they are the main propagator in that, so that we can start new cycles and end old ones.
Yeah.
What came first, the death cycle or the mushroom?
That's a question for God.
Fair.
So I guess the next question is a little bit about like the whole cycle of a mushroom. So like, can you explain a little bit about like what happens in its life cycle a little bit, from spores to end of life and decomposing and so on?
Sure. Yeah. I mean, like from the scientific perspective. um, so I don't know where you want to pick on the circle that we can start, but maybe we'll start with the mushroom because everyone can. What do you mean? You mean like the you're looking for like a prescription for a specific type of mushroom?
Are you talking about psychedelic mushrooms? Or are you okay?
Yeah, like excessive, but like so she had asked for anyone that didn't hear, for someone that has never done mushrooms, psychedelic mushrooms, what do you recommend?
So, yeah, I make supplement. This is probably something that we'll actually touch on a little later, but I mean, why not touch on it now? I make, I guess, supplements and also microdose capsules. And the main thing with me is, I don't know, I live in New York, and anybody that talks to me about mushrooms is like super intense and wants to take just like a 3-5 plus to the face. And I'm like, okay, can we just relax and get to know the spirit of this mushroom a little bit and like build a relationship with it? Because there's all different types of strains and there's all different types of like, communications that each individual strain, and even within the same strain, each of those mushrooms is trying to tell us and like use this as a channel. Like that's just, I guess, my personal belief. But I think that is really nice to microdose, like, and even everybody across different communities, there's different definitions of like what even is a microdose. But there's some organizations that are clarifying what that is, and that's also like what a microdose is also going to depend on the potency of the mushroom. So the amount of like milligrams per gram of the psychoactive compounds, which might be like psilocybin and psilocin. And there's all different ways that like, yeah, we metabolize those compounds, and there's like more to say on that conversation. But it's really important to like, if it's accessible, to like try to get your mushrooms tested so that you have an idea of potency. Because like there's two mushrooms that might look exactly the same, but one of them could be 5 times as potent as the other one. And so that's crazy. So anyway, like in either way, in any case, just trying to take a really, really, really small amount, like take 0.05 grams of it, take zero point 0.1 grams of it.
Currently, no. The current evidence suggests that you shouldn't need to be any corrected dosage for body mass index or weight or anything of that nature. Although, I mean, that could be subject to change. But in ge- yeah, yeah, it doesn't seem to be the case with at least with like the breakthrough dose of psilocybin. Um, but to echo Emily's point, like with microdosing and getting familiar and getting what do you call it? Like, yeah, I guess just familiar, affiliated with like a new substance. Starting small and working your way up is always like a safer. I don't quite. It's possible, and everyone's different, and not only just in your body type, but in your brain chemistry type and your genotype and this sort of thing. So, definitely, like with mushrooms, less is always more, and starting with someone that can help you get a targeted dose as you learn would be really helpful.
Yeah, the people in your community that are cultivating and making products and like interfacing, um, it's important to ask them those kind of questions about dosing of like, do you know the potency of your mushrooms, and what is the dose? Like, how can I make sure that I'm properly dosing myself? Like, what have you done to ensure that there's like homogeneity in your chocolate or things like that? Um, yeah, so these are definitely questions for that. Yeah.
Awesome. So yeah, it jumped kind of into like the supplementation aspect of it, so that's good. Oh, I was gonna bring one last question about the last topic was if you want to talk a bit about what breeding of mushroom would be. Um, uh,
Yeah, we will have a Q&A.
Yeah, we will have that for sure.
yeah, we'll take questions, I guess, more at the end. But we're gonna go through just a little bit of the topics, and then at the end, hopefully, we answer some of your questions. Um, but yeah, again, we'll try to ask a bunch of questions at the end. But yeah, if one of you guys want to talk a bit about like breeding and what that is, what's like, how would you do that? You just take two mushrooms, put them together, and they go at it, or what's up?
You want the birds and the bees?
Yeah,
The
tell
whole
me the
thing?
birds and the bees story.
Well, I always think it's interesting that when mushrooms meet, when two spores meet, it's very romantic. Um, they have a candlelit dinner, and they make a decision on whether or not they're going to share space together. And if they do decide to share space together, like one has the right perfume on and the other one chose the right cuisine for dinner, they do move in together and they cohabitate a mycelial network where basically one moves in with the other one. And uh, they share and sort of do this dance in the mycelium, and then they eventually do their propagation. They form the mushroom together, and then on the face of the fertile surface is where they have sex and produce their children, and stuff like that goes on. So for breeding of mushrooms, to get back to your question, is like intentionally having a sort of Cupid-like role in this process where you're like sort of matchmaking and you're taking one mycelium from one and mating it with the other, and letting them form a mushroom together. And that first, like I said, they move in together, so that actually has that breeding hasn't happened yet until the mushroom forms and they actually make the spore. And the spore is now actually the hybrid, and that first child is now the cross. And so, I could go on and on about, progeny and isolating
Right.
traits and stuff like that.
We
but
don't need to get into
yeah.
too deep, but yeah. So this just happens on like a log outside, is what you're saying?
This happens on a log. They find them. They find each other, right? I mean, they have like signals and they eat stuff and most. I think the most common instance is they interbreed, because when you think about a mushroom, it's shooting spores and they're dispersed by wind. And if you look at the probability of a spore going a certain distance, it is like exponential decay, meaning that like for every inch that further away from the mushroom you are, it's 10 times less likely that one of your children is going to land there. So a lot of times they're interbreeding.
So I can make, I could make like a psilocybin oyster, is what you're saying? I could cross like two things like that.
No, they have certain barriers, just like how you can't go and make babies with donkey. Yeah, can't.
I feel like it would be interesting to hear like from the cultivator's perspective, because like for psychedelics at least, you'll run into like people are starting to crossbreed so many different strains. So it's like the cultivator I work with, he has all these crosses, and I like even me not being a cultivator, like I'm not even really sure what that means. So like Steak, or I don't know if you could comment on that either, but like what from a cultivator's perspective, why would you wanna cross two mushrooms? And then how does that actually look like in a hands-on way?
Yeah, I'll mention it briefly. I'm just delving into it, so I'm not like crazy fresh on like terminology and shit like that. But as far as like why like I want to do it is partially just because I want to learn, and also, but from like a grander perspective, like you can also like reinvigorate genetics. If we're talking about like excessive inbreeding, that might have implications down the line. So maybe we want to back cross fresh genetics in there and reprogenate and create a new culture with higher virility, or just like higher strength and better equipped to kind of take on the world. Like think about shit like if my family was inbred for fucking 10 generations, I'd probably be a way different person. So maybe we want to insert something new and fresh. And I think even just like on the creative aspect, it's, that's like a background information for my head. It's more so like, oh, now I get to learn something new about mushrooms I never knew about, and I think that's more so potentially for a lot of people where that arises from is just the kind of creative aspect of like, oh, what can be done? What could we do? What could we try? And, incorporating new aspects and techniques into things and just having fun with it. I guess from like a nuts and bolts kind of situation. And I think there's also like a tricky thing within the community too, is like a lot of times a lot of these crosses aren't well documented either, and a lot of people are just throwing names on shit, and I don't even think it is a cross. So that also becomes confusing, and I don't think there's enough. I think it's getting better because there's a lot more people that are like trying to perpetuate that, and obviously like as the regulations and things change, like the proof should be there. And sometimes it's hard to just like take someone's word at it, especially with a lot of I guess like rudimentary systems of just kind of mixing spores and then selecting traits that appear to be crossed. You're never going to have a confirmation that what you did is correct, and it could just be, what do you call that, spontaneous mutation or spontaneous what's the proper word? Phenotypic expression, just something spontaneous of like, boom, this happened now. So I think for me, like trying to do things the proper way and develop a monokaryon, which is like what Jordan was talking about, is a mycelium that hasn't made room and cohabitated with another one, versus a dikaryotic mycelium, which has done that. And properly attaining a monokaryon to then mate with another would be a way to actually confirm what you're doing is correct. But yeah, I guess to answer the question more, for me, it's all just creativity and having some fucking fun.
What about the differences in like?
Oh no, I was just gonna agree with you, bro, for sure. I think for me, the reason why, or my interest in breeding, um, is like looking for certain characteristics. It's just really fun to like try to work on a project and then have an end result. But mushrooms, it does a lot of things. But we're gonna have a Q&A at the end, and I think we'll be able to answer a little of those questions. But no, ultimately, yeah, shit is just fun. Um, but I think yeah, it's important to always, um, you know, maintain like the land races and whatnot, and um, yeah, just make sure those are good too when you're doing all the crossing and whatnot. In my opinion, but I guess that's a whole another topic. But
So there's like a whole lot of different mushrooms out there that are active, right? Like, are there different like experiences with all of them, or what's the difference with like I don't know if you guys saw the Zapaticorum and all the different things Stieg is cultivating. Are there like different experiences with each one?
I would say yeah, big time. I mean, there's different compounds in them, if I'm not mistaken. I'm more familiar with psilocybin cubensis, or psilocybe cubensis. Um, but yeah, and yeah, there's definitely different effects you get from these mushrooms, even within um, that realm of mushrooms. But I mean, I'm sure you guys could speak more on that, on the different effects from like exotics and the compounds and whatnot.
Stieg, do you have anything to add on that?
Uh, yeah, I think there's like a pretty significant difference. Now, I've never like blinded myself and tried to see if I could pick them out, but I honestly don't really care to do that because I have my own belief and I'm going to stick with that because I like this reality currently. But I think like on a certain level, like you could look at it like how Tony's saying, like there is different compounds, there's different things like that, but also in cultivating different like species of mushrooms, you have to develop a different relationship with them, which has an impact on the experience as well. So I think psychedelics as a whole, like even beyond just like the chemical constituents, having it be so personal and so ineffable, it's like we're always going to have some major impact either from the chemical constituent or from the environmental constituent or from the relationship constituent. And part of that kind of bleeds into the aspect of like a lot of kind of exotic mushrooms where we're working with currently tend to be more from the wild, more land race, and there's less kind of domestication and like breeding being done, so we don't have as much human influence within there. So I think we also take in messaging from the specific land that they come from. I like to think of to kind of filter back into the decomposition aspect of mushrooms. I view that as a translation. So mushrooms are master translators. They take in from the natural, retranslate it into something to be spoken back. And so when we take a mushroom or talk with a mushroom or hang out with them or whatever the fuck we want to do with them, they're taking all that information and reconjugating it into a new form of conversation with us. And so from that perspective alone, a lot of these mushrooms come from very specific or different places in the world. And then it's like, boom, what does that place have to tell us? What do those people have to tell us? What does that ecology have to tell us? And those mushrooms can enact that and express it through their form and their experience and whatnot. But that's getting into maybe some woo woo hippy dippy type shit. So
I don't think it's woo at all. I think that if you're out and foraging and you do find, like, I guess it's kind of leaning toward, the conversation is leaning a little bit towards psychedelic mushrooms in my head, but it doesn't have to. Like, if you find a cubensis out when you're foraging and you take it from that environment and you get the genetics, which can be done in a couple of different ways, but you pretty much like would get the spores in order to make some type of like liquid culture that you can use to start cultivating that mushroom. But like the second that it's in your energy field and like you're working with it, you're already going to be influencing like what that mushroom is going to express like. And then that conversation gets even deeper when you're talking about like what type of substrate you're using and like what type of conditions and how am I like, what kind of nutrients am I feeding the mushroom? There's like all of these steps that's gonna influence it. So like, yeah, every single time a mushroom grows is different, even if somebody's telling you that like this strain and this strain or whatever, like this strain is that. And as far as like, I guess what testing looks like, because there's definitely like, I really appreciate Stieg what you said, because it is so multifaceted, like what the relationship with mushrooms is. It's like there's your spiritual relation with it, there's the relation to the cultivation, there's so many aspects, but from one scientific perspective, which is like the whole testing element, you can pretty much test mushrooms for their array of different compounds. So like I mentioned before, there's psilocybin and psilocin, and there's also these other compounds that people are looking at, like baocystin and norbaocystin. And what people that are studying these compounds are finding is that each of these compounds might be responsible for some aspect of your experience with the mushroom. But like I was saying, there's so many, it's not just like, it can never be pinpointed to one thing, like, oh, this mushroom has so much baocystin, so I was having so much visuals. Like, maybe that will be part of it, but it's always going to be more than that because it comes to like the personal relationships and the chemistry. But as far as like what that testing looks like, people are categorizing mushrooms on kind of like a spectrum according to their compounds. And I don't know, that's another conversation of like what that testing looks like.
That's the next question right there. You already kind of got into it a little bit. But how can we use some of those testing agents that you're talking about for like harm reduction purposes and that sort of thing?
Yeah, so I think harm reduction and just like more informed dosing is the main reason why I'm interested in it, not so much to try and like categorize mushrooms into being like this or that. I think that, so pretty much, I guess what the testing has looked like so far is we use HPLC testing, so it's high performance liquid chromatography. And using that machine, we test extracts that are made from the different mushrooms, which there's been a lot of like cordyceps testing, so you can test cordyceps for cordycepin and adenosine content, or you can test psychedelic mushrooms for those other compounds that I mentioned earlier. And that's a whole conversation of just like what are we actually testing. But there has been a lot of citizen science and community science work to develop and refine the protocols for mushroom testing to understand the concentration of the bioactive compounds. So by that, I'm saying like how much cordycepin is in this cordyceps mushroom, because you may be trying to take like an amount in a specific range to address a specific issue that you might be having. So like cordyceps can address inflammation, for example, and can help to oxygenate your body, but it will do more or less of that depending on how much you take. So it's really about informed dosing in that sense. And with harm reduction, it's the conversation that we had addressing that person's question of like, how do I know how much to take? Well, yeah, that's actually a really hard question to answer if you know nothing about the potency. And depending on people's background with mental health or whatever else, you want to make sure that you're starting low and like not having too much too fast, because there's so much that goes into like dosing with psychedelic mushrooms that's important, like setting and building a relationship with the mushroom. But sometimes like that's a journey for everybody, that's personal, and everybody's learning kind of as they go sometimes, as we are developing more community spaces around it. So the testing aspect helps people, I think, to get a better foundation for what they're working with. But it's still, I would say it's not to the point yet where there's like a lot of accessibility in testing, because sometimes there's price barriers for cultivators, or it's just like, I don't know, you might not be tapped in with testing yet. So, yeah.
Yeah, well said. I
I
guess
have a
that kind
little-
of correlates in.
I have a little piece to add to
Yeah,
that.
yeah, of course.
Because I also think with the testing, it gives, like, from my perspective, it gives another level of agency to me. Because like I've had a lot of experiences when I was growing, specifically lots of different paneolus and giving it to friends and trying to tell them like, yo, please do not eat a lot. Like you only need a little. Like these are super strong, and they'll be like, oh yeah, my other dealer says so he's got the best shit too. Fuck you. And he'll eat a whole jar, and then I'm just like, dude, like I told you. And so I think it's helpful to be able to have some sort of like specific hard data that you can make a comparison and give credibility to your word, because some people don't even want to believe what you say.
Yeah.
And that can be super dangerous.
Exactly. And what the hard data looks like when we go through these testing classes, which we do it with the Center for Mycoanalytics, but it's also other testing labs that produce this kind of data. The end goal so far has really, there's a lot of different things to study as far as like experimental design wise, but the end goal so far has been, okay, I want to know how much milligrams per gram of this compound is in this mushroom. So then you have like, say you have a mushroom and it turns out that there's 6 milligrams per gram of psilocybin and then like 2.2 milligrams per gram of psilocin. You have an understanding of like how much psychedelic bioactive compounds are in there. So you could be like, all right, if I eat 1 gram of this, like that's how much of these compounds I'm taking in. And then when you start to like compare the mushrooms laterally, it's like, yeah, you might get one from Stieg that's like 35 milligrams per gram. And so it's like, that's on the agency that he was talking about to be able to properly dose that. But that's also a community stewardship thing of being like, we need to have more of the testing done, you know?
Yeah, definitely. I guess one of my next questions is a bit about the supplement industry and how. Did you want to say something?
I was just going to add real quickly on the harm reduction side of things. In the most extreme instances that I've seen, one mushroom could be 40 times stronger than another mushroom. So, yeah, testing really provides a lot of agency, and I think we're going to see a lot more accessibility with future developments in technology, and more people hopping on the board, and maybe some social services to help support harm reduction efforts in psychedelic spaces.
Definitely. So as far as like I guess the supplement industry goes, are there any things that I should be like look out for in correlation maybe to like mycelium versus fruiting body, or like what does that even mean? Can any of you guys elaborate a little bit more on those topics?
I was talking to some people outside about this earlier too, about mycelium versus fruiting body. Yeah, so the main argument is that certain people are using mycelium as a cheap production method to produce supplements for the masses. The mycelium producers themselves are saying that's not our intent. Our belief is that we've done research, and the compounds that we are interested in getting out to people are actually produced in the mycelium, and they're specifically produced when the mycelium is grown on this media. So yes, while it may look like our products have a bunch of filler in them, we need that in order to make this chemistry. And so it's really convoluted, and there's a lot of like marketing that and money grabbing that kind of further muddies the image. So it's hard to get clear resolution, but what I like to consider is that these are different parts of the life cycle, as we were talking about earlier. And the fungus is producing different chemistries in different times of its life to be successful in that part of its in that chapter of its life. And with curated and intentional use, this chemistry could be applied and be beneficial, whether it's from mycelium or fruiting body. So I don't think it's appropriate to say one is inherently good or bad. It's more about how it's curated and what the application is and how you're dosing it and where you're bringing it. And really being informed about what you're trying to do.
And that's on like from an education standpoint, it's really about asking the right question to the cultivator and like learning how to ask those questions to pressure the cultivator to have more transparency behind what it is that they're doing. So yeah.
Awesome. That sounds. Yeah, I agree. The next part of it is a little bit about DNA sequencing. Um, like what is I hear people say DNA sequencing, and I don't know what that means. Can we explain a little bit about what that is and why it's important, and some of the aspects of maybe going out and finding a mushroom and trying to identify it?
Well, DNA sequencing is at the core principle is reading the genetic code, the DNA, of any given organism. But most commonly in mycology today, what people are talking about is DNA barcoding, which is reading a small segment of a mushroom's DNA to identify it to a certain species level. And so this is what's being done at like by mycologists to better understand mushrooms in our environment and across the world, and to have a better understanding of their diversity, individuality, or what is shared. But further advancements in DNA sequencing, there's all sorts of applications that you can do with reading the genetic code, whether it be, learning about a mushroom that does something novel, and learning the genes that are responsible for doing that and applying that. There's a lot that can be done with the technology.
Yeah, that's one thing. And then also the DNA sequencing and DNA barcoding. What I find really interesting is trying to get this to paint the picture environmentally of like what fungus is existing where and when and why. So it really helps us understand our environment, because also our environment is changing so quickly, just in terms of like the amount of moisture that's available in the environment and the amount of like heat and just the actual biodiversity that's shifting in the environment of like things are or are not existing anymore. So I haven't really gotten to delve into any of this with my work, but just like you could use DNA sequencing or DNA barcoding to like survey an area. And I think it would be really cool to like do that over the span of 10 years and see like what organisms are still existing here or not. And that's kind of like, I guess I'm kind of stretching a little bit away from like just DNA barcoding and sequencing, because there's ways to take reads of like all the different organisms that are active in an environment. But then there's also just like you could go out and forage for mushrooms at the same place for like 10 years and sequence them and things like that.
Yeah, I mean the eDNA, environmental DNA surveys are really interesting, especially in like stuff when you're doing land surveys and stuff, and you can track populations of organisms and how they're responding to like certain environmental stressors or something, maybe like a pollutant or something like that.
Are people still finding new mushrooms? Like, could I discover one if I went out and looked in the forest?
Yeah, if you spent enough time, that's the thing, is most people discover new mushrooms every single day, but they don't take the time to tell anyone about it. And they don't actually tell themselves, because they don't realize that they're looking at a brand new mushroom. But almost any time you go out in the woods, and I bet around here, and in kind of like an underserved area possibly, as far as like mycologically, there's probably lots of new species or species that are going under their European name that were just assumed to be the same that are actually unique to here. And then, like how we were talking about earlier, how like this year has been a particularly wet year, you might find a mushroom that only pops up once every 40 years or something when the conditions are right. So every day is an opportunity, and I think it's a really ripe time in mycology with the accessibility to DNA barcoding and yeah, and social media too is really powerful. There's like a lot of free education sources.
I think that DNA barcoding and sequencing is really important because, like, for me, taxonomy is so fucking confusing. Like, it like changes and it's influenced by who it was established by, and that's when you're talking about all the like the Latin names and everything. So it's kind of, you can kind of push through that a little bit by just getting the actual like DNA read of a mushroom. And just back to that one thing that you said, like personally as an environmental scientist, I work in soil remediation. So like one question that everybody always asks me is like, oh, well, what mushrooms can we use to like remediate the environment? And that's a great question. And there's definitely been research about it. But another application of DNA barcoding could be like, you could go to a site like Jordan mentioned that has been polluted. Like I work in like oil polluted sites down in Texas. And so you could go to a site and be like, okay, well, what organisms are actually growing here and like how are they active in the environment? Like, do they actually have the capacity to break down these certain compounds that are harmful? And then you would want to study like the genetics and the DNA of that type of fungus to be able to like take action on using that as a remediation tool.
Yeah, from my perspective, I think DNA barcoding and sequencing is super sick, especially when you get into areas that where mushrooms are familiar, where mushrooms aren't that familiar. For example, I just came back from Africa and I was in like a spot where people really don't know what mushrooms are. And with something like DNA barcoding, we would be able to help them identify these mushrooms and like actually properly like let them know like this isn't going to kill you. And then a lot of the times, we're talking about discovering new mushrooms. When you go to these remote areas, a lot of these mushrooms haven't been tapped into yet. So you're able to really just educate people of the land. It's not really even educating. I feel like it's more so like reminding them because a lot of that shit is lost knowledge. But helping them get back to their knowledge. So that's my perspective on why it would be beneficial, just helping people remember some info.
Facts. That's really important about what he said about the like fungophobia, or what is it, mycophobia? There's so many cultures around the world that have developed like a fear of mushrooms. So like, yeah, as a reminder or as like to affirm people. What's the right word? Just to make people feel more confident in what they're eating and engaging with.
So the next question is, if I were to go like out in the woods and I wanted to ID a mushroom, like what do I do? What are the steps it takes to do that? Are there any like resources or tools I could use to do that? Should I pull up my AI phone and just try to ID it that way, or is there better methods than that?
Yo, straight up, there's a bunch of cool like mushroom identification books at like Barnes and Noble online and whatnot. If you guys are living around here, a great mushroom identification book is Mushrooms of the Upper Midwest. It just narrows it down to mushrooms that are in the Midwest because there are a bunch of mushrooms out there. And then if you wanna, and there's also apps like iNaturalist that's really sick. It helps identify biodiversity around you. And it isn't AI. It links you to like experts and whatnot to help identify the mushroom. And then if you want to take it a step further, you can do things like spore prints and tissue cultures, and you could actually just try to regrow it, which is fun. But yeah, straight up. I think that's how I would try to ID some mushrooms.
Yeah, for sure. Definitely tap into your local books. And a lot of the times in the beginning of the books, there's like, it will walk you through all the different parts of the mushroom. Because when you're out foraging and looking at mushrooms, like, there's so many different aspects of it to study. And like, some mushrooms, like, very clearly have different shape or morphology, but then some kind of are lookalikes. And then you're like, well, what would be the defining trait that would differ between these two? And like Tony said, you could definitely do a spore print. And some books, I don't know if the book that you mentioned does it, but like some books will tell you like how to do the spore print and be like, oh, the spores of this mushroom are going to look purple or brown. So you would have to wait, some time before you could more confidently identify a mushroom. But also, it's great to have pocket guides when you're out foraging, and also taking pictures of it, and not just taking picture like, oh, I saw a mushroom, but like taking a picture so that you can actually see like, what do the gills look like, what does the shape look like how big was it, what else was growing around that mushroom, like was it growing on moss, was it growing on a log. The environmental factors are really important. And then like bless you, the progressive steps is like, okay, now that I took these pictures and have these key like notes about what this mushroom is, I could bring it to a community space and be like, has anybody else like ever seen or encountered this mushroom? Because it's always good to get multiple, especially if you're foraging to like eat. It's always good to get multiple like checks after you forage and be like, this is what this is. And sometimes that looks like a Facebook group because there's a bunch of knowledgeable people there, or just like straight up the people in your community.
I think another really good thing to do as well is don't eat the first one you find. And like try and find it in multiple locations and find it in multiple environments and wet, dry, so you can get a feel for all the different kind of morphological changes that'll happen based off environment. Because sometimes you'll have a mushroom that's the exact same, but since it's growing over here and it's a little bit drier, it might look totally different. Um, it might look like something else. So it's good to get a feel for the breadth of how these mushrooms can look in the wild, because they're not all going to look exactly like the picture in the book, and getting familiar with that. So I like to use a rule of like 5 or 10. I like to find it 5 or 10 times before I even potentially engage with it. But that's just my own personal rule.
Yeah. Should we do the Q&A? You guys have any questions out there? Yeah.
So I had a question about contamination and propagation speed of mycelium. So I'm just kind of thinking about the thing you talked about with looking at breeding different species. There's two different strains that become friendly and they like each other, then they have plenty together. Is there any thought to different substrates, whether they're sterilized or pasteurized, and what on the substrate changes how quickly like mycelium would spread and propagate on there? So, for instance, if there's friendly bacteria or something that works in the best interest of the mycelium, maybe it's more ready to spread quicker versus something that's more hostile. So think of it as like if you're putting something entirely on first, and then you put the on top of that, and it keeps
I'll say just a little piece. I think as far as like pasteurization versus sterilization, there's not going to make like a huge difference, in my personal opinion. I'll utilize like we can talk about like beneficial microbes and things, but like how beneficial are they really being? And I tend to look at the beneficial microbes as more of like an immune system for the substrate, more so than like a symbiosis with the mycelium itself. Now, could there be situations happening? Probably. And I look at pasteurization as more of like kind of like an efficiency mode. That I don't have to spend so much energy sterilizing everything, and I can get away with pasteurizing and do a lot more at once, where sterilizing will take me a lot longer and a lot more energy. But maintaining those microbe levels, I don't want to say that there isn't that there isn't inherently a benefit to it, but I think it has more to do with just creating an already colonized substrate with something that isn't a hindrance versus something that's a benefit. if that makes sense.
And just one thing, I've had some experience with different, like working in petri dishes and having different what's the jelly called? Agar, with different agar recipes. And from that, I was able to see, because there's like so many different agar recipes. So if you're curious about a certain cross, you could you could isolate the spores from a mushroom and then have them grow in the petri dish and see, like, feed it different agar and be like, what makes it grow faster and what makes it grow a little bit slower. And when I broke it down to that perspective, I was able to understand more of like, okay, well, this would feed into a certain type of contamination versus this wouldn't. But you might need a little flow hood for that.
Anyone got any other questions out there? Yeah.
By types, I'm going to guess you mean species, different species of mushrooms. There are interspecific hybrids of mushrooms. Yeah, they're pretty new. I think one example in the gourmet world exists. The black pearl king oyster I heard might be an interspecific hybrid.
Mm-hmm.
And then there's a couple psilocybe mushrooms that are interspecific hybrids, and then there's maybe a couple paneolus mushrooms that are interspecific hybrids. I haven't grown any of them personally, but I've seen pictures of them. They're cool looking.
And there might be like some good research papers out there about it.
Yeah, there might be some good research papers about it, and it's also kind of new and evolving. Our understanding of species within the genus is constantly evolving. Like, for example, we had on the west coast psilocybe cyanescens, psilocybe azarescens, psilocybe olenii, and they were all distinct species related to psilocybe subruginosa in Australia. But now there's an Australian mycologist that's proposing that they're all the same. So, and they all do interhybridize and stuff. So, but they just look a lot different. So, um, it's weird. There's so the question was, is there a any data on shelf life of mushrooms as far as best way to harvest, process, and store them, and this sort of thing? There's lots of ongoing studies currently between both academic labs and private interests and citizen scientists that are exploring, specific questions within those. I don't think there's like currently a well accepted and agreed upon best methods and practices for optimal shelf life of mushrooms. There was one paper that came out that had said that they degraded fastest at colder temperatures, but a lot of people weren't like finding this in their own personal experience. So it's kind of all over the place. My personal recommendation from what I've found is to store mushrooms dry as possible. Moisture
Cracker dry, they call it.
Yeah, extremely dry, like with desiccant. Lots of moisture absorbers, and the moisture absorbers that are color changing when they get wet or there's like an increase in moisture. Those are really helpful for you to see, that they are soft inside. And then the proper storage vessel. So like glass is non-permeable. Like plastic is okay, but it does permeate a certain, like it's very like microporous over time. So moisture and air does permeate over time, but glass is like our best thing. And then if it's dry and it's in glass, then frozen is ideal. But without those first two, if you freeze mushrooms, you can freezer burn them, just like with other foods, and they'll degrade that way. So, yeah, I'm happy to talk to you about it more specifically after if you're interested.
And also be careful of like the freeze thaw thing, because that can like introduce more moisture into there.
Totally, yeah. Like, yeah, if you do store your mushrooms in the freezer, in like a bulk jar, take it out of the freezer and let it come acclimate completely to room temperature. Because if you open it while it's cold, it's going to act as a condensing surface, and water in the atmosphere will condense on your mushrooms and freeze, get freezer burn and rot. So either let it acclimate to room temperature before taking out a serving, or store them in serving sizes. So, but yeah.
Wait, and this is a random thought that I had earlier that I meant to say about foraging and identification books. But yeah, there's apparently more AI generated mushroom identification books that is apparently causing people to die. So, like, please be careful and get reputable reference books. Yeah.
If you didn't quite hear him, he said that there's the Minnesota Mycologic Society that you can join. It's $50 a year, and they'll take you out on foraging journeys and help you identify some things if you want to get started. I had one last thing if you guys want to touch on.
I'll just touch on the Mycological Society. It's a great resource to get involved with your local mycological society. Since I got involved in mushrooms, a lot has changed, and it's maybe not as mentioned as much because of resources like iNaturalist and because of social media. You can be in any mushroom club on the planet on the internet, but before that, it was really joining your local mushroom club. And these people are very knowledgeable and love to share everything they know. They're really passionate. They're really like truly into mushrooms for mushrooms. I would just be, respectful and stuff. Like a lot of these people may not be like privy to the psychedelic aspect of mushrooms. A lot of people may encounter that are actually like really mushroom people. So just be respectful and stuff, and yeah, when you're in those sorts of spaces.
Oh, my last question for you guys here is heard of this thing called woodlovers paralysis. Well, what is that? And is that something we need to be worried about? Like, what? I don't know what that is.
I'll touch on it quickly. It's a side effect of a certain species or group of species of mushrooms that causes temporary, partial, or full body paralysis for a period of like 2 to 12 hours. And it only happens with a certain couple species of mushrooms. So if you're not making any plans to trip in New Zealand, Australia, or the Pacific Northwest, you probably don't have to worry about it. But if you do, yeah, this is a side effect that can happen specifically with Psilocybe cyanescens and related mushroom species. It is transient, and it's not currently nobody has like fallen ill from this. Besides, it's kind of like scary. I think just spreading awareness of it and making sure people understand that there's a possible side effect of mushrooms.
I'd like to ask a follow-up because here in Minnesota, the access to the cyanescens, azarescens, and those types of mushrooms is pretty limited. There might be a handful of people growing them, but overall, it's not something we will generally encounter here as far as we know. But on the cultivation end, I'm curious if there's any information on whether or not substrate is impacting the cause of or increasing the prevalence. I've heard stories of like substrate being a potential cause or even age of the mushroom being a potential cause, or do you think it's purely genetic or random?
Currently, well, the chemical agent that causes it is not known, so it's really hard to start asking the questions beyond that because we don't know what it is. And there's some good ideas and some candidates like that, people that are interested are currently chasing. But from lore that I've heard, kind of to go with that, is like certain patches consistently give it to people and other ones don't. Or certain people are more likely to get it than others. Um, and some people have suggested maybe it is like substrate based. It happens in wild ones. It happens in cultivated ones. So it definitely seems to be like not super hyper specific to substrate. Like it could be produced, this chemical or whatever causes this phenomenon could have a couple different inputs and still have that output. Um, but it seems to be mostly with high doses too. So I haven't heard of like anyone microdosing cyans and having paralysis. So some of the variability might also be tied to the variability in potency. That's just like, you know, apparent in mushrooms.
Well, you guys did great. Thanks for answering all the questions we had. Does anyone have any last words that they want to say up here? No? Any other questions in the crowd? Yeah.
So just a question on visual characteristics versus properties of mushrooms. So I've heard that if you see luminous in a mushroom, that's an indication of the level of psilocin. But like when you look at ape versus pea bean, what are the albino characteristics have to do with mushrooms? Like any insight on coloring and how that affects the body of mushrooms?
So we do actually have to wrap up. I think everyone will be maybe open to connecting afterwards if you want to do some one-on-one questions. But we gotta wrap up at this point.
Thank you, guys.
Thank you, everybody.